So let's begin chapter 24, the origin of species. So when Darwin explored the Galapagos Islands, he discovered many plants and animals that were found nowhere else on Earth. Now let's define some key terms that you need to be familiar with. First, speciation is the appearance of a new species due to biological diversity. So differences can be good. And speciation is the focal point of the evolutionary theory. Now, the evolutionary theory must explain how new species came about and more specifically how a population has evolved. Now, we have microevolution, which we talked about, consists of changes in allele frequencies, while macroevolution is evolutionary change above the species level, which is just a fancy way to say the formation of a new species. So let's define what it means to be a species. So species is a Latin word that means kind or appearance, and species is a grouping for organisms. Now, biologists compare the morphology, the physiology, the biochemistry, and the DNA sequences of organisms when they are grouping them. Now, the biological species concept defines a species as a member of a population or a group of a population who are able to interbreed and produce viable, fertile offspring, and they are unable to produce viable, fertile offspring with members of other species. So it is simply defined as a member of a population who can breed and produce fertile offspring. Now, it is key that the offspring be fertile to be considered a species. For example, a donkey and a horse can breed and they can produce an offspring, but it produces a mule which is sterile. So they are separate species because the offspring, the male, is sterile. Now, the biological species concept is based on the potential to interbreed rather than physical similarities. For example, you have lots of diversity within the same species, such as within the human population, but the ability to interbreed and produce fertile offsprings makes them all the same species. So we can all have lots of diversity, okay, but the ability to interbreed makes us the same species. Then, on the other hand, you can have similarities between the different species that cannot interbreed. For example, the meadowlarks look alike, but they cannot breed. So why don't different species have the ability to mate? This is because of what we call reproductive isolation. Reproductive isolation is when biological factors or barriers prevent members of two species from producing viable, fertile offspring. Now, reproduction isolation is influenced by a combination of various reproductive barriers or factors. Reproductive isolation can be classified on whether the factors act before or after fertilization. An important definition to know when we're talking about reproductive isolation is hybrids. Hybrids are the offspring of crosses between different species. So there are two types of barriers that create reproduction isolation of species. They are prezygotic barriers and postzygotic barriers. Remember the prefixes, pre meaning before and post meaning after. So our prezygotic barriers prevent different species from mating to begin with. They are barriers that happen even before a zygote can be formed. Then we have postzygotic barriers, and they happen if a zygote is formed, and they often prevent the zygote from developing into an adult. So here are all examples of prezygotic barriers which prevent mating before a zygote can even be formed. So prezygotic barriers stop fertilization from occurring in different ways. First, the barriers prevent different species from attempting to mate. Next, the barriers can prevent the successful completion of mating. And last, The prezygotic barriers can hinder fertilization if the mating is successful. So let's go ahead and briefly talk about each example. The first prezygotic barrier is habitat isolation. Habitat isolation is when two species occupy different habitats so they encounter each other rarely or not at all. For example, the two snakes in the picture live in two different habitats, so they can't mate because they never meet. Then there is temporal isolation. This is when the species breed at different times of the day, or different seasons, or at different years. So one species may come out in the day, and the closely related other species comes out at night, so that they can never breed, or one is hibernating while the other is active. The next is behavioral isolation. Behavioral isolation is when species behave so differently that they do not mate. So, for example, the courtship and other behaviors are unique to the species, such as songs or the mating dance. Then there's mechanical isolation, which is when the morphological differences prevent successful mating. And the last prezygotic barrier is gamete barriers. Now, gamete isolation is when gametes can't be fertilized. So it's when the sperm of one species is unable to fertilize the egg of another species. Then, if the different species do mate, there are different postzygotic barriers that cause different variations of reduced viability of hybrids. Now, postzygotic barriers essentially cause the offspring to be sterile and cannot produce further offspring. So let's talk about the postzygotic barriers. The first postzygotic barrier is reduced hybrid viability, which is when the genes of the different species may interact and impair the hybrid's development. Then there is reduced hybrid fertility, which happens when the hybrids are vigorous, but they are sterile. Then the last postzygotic barrier is hybrid breakdown, which happens when some of the first generation hybrids are fertile, but when they mate with another species or with either the parent species, the offspring of the next generation is sterile. So the biological species concept is the most common definition for a species, but there are limitations to it. First, it cannot be applied to any organisms in which we do not know about their reproductive choices, such as asexual organisms or fossils. Also, the biological species concept emphasizes the absence of gene flow. However, gene flow can occur between distant species. For example, the grizzly bear and the polar bear can mate to produce a growler bear, which is a hybrid. So there are other definitions of species that emphasize the unity within a species rather than the differences. Other definitions of species include the morphological species concept, where they are different based on how they look. Then there is the ecological species concept, which says different species occupy different ecological niches, so that makes them different species. Then there's the phylogenetic species concept, which says that species have the same genetic history in the phylogenetic tree. So remember, we define speciation as the appearance of a new species due to biological diversity. Now, speciation can occur in two ways, in allopatric or in sympatric. Now, allopatric speciation is when a small group gets isolated from the parent population and a new species is formed. And in sympatric speciations, a new species occurs within the population in the same place. Now, SYM means the same, so it means the same location. There's no geographic separation. There is some other type of reproductive isolation. So once again, allopatric speciation is when a small group gets isolated from the parent population and a new species is formed. Once geographic separation has occurred, one or both of the populations may evolve independently through mutations, natural selection, and genetic drift. Now, reproductive isolations may arise as a result of the genetic divergence creating new species. An example of allopatric speciation, or when a population is divided into geographically isolated subpopulations, is the snapping shrimp by the Imus of Panama. There are 15 pairs of sibling species of the snapping shrimp that are separated by the Imus of Panama. Now, these species came about 9 to 13 million years ago when the Imus of Panama formed and separated the Atlantic and Pacific waters and thus separated the subpopulations. So here is the Imus of Panama. It is a narrow strip of land that lies between the Caribbean Sea and the Pacific Ocean, linking North and South America, and contains Panama and the Panama Canal. And it separates the different snapping shrimp species. So the allopatric speciation occurred in the snapping shrimp populations. So geographic barriers create allopatric speciation. So regions with many geographic barriers typically have more species than do regions with fewer barriers. Now, reproductive isolation between populations generally increases as the distance between them increases. For example, reproductive isolation increases between dusty salamanders as they live farther apart from each other. Now, it is important to know that isolation barriers to reproduction are instinctual, and separation itself is not a biological barrier. A laboratory example of allopatric speciation was performed by Diane Daub, who raised two populations of flies on different mediums separately, one on starch medium and the other one on maltose medium. Now, after many generations, natural selection resulted in divergent evolution. Populations raised on the starch digested starch more efficiently, while those raised on the maltose digested maltose more efficiently. Dobbs then put the flies from the same or different populations in mating cages and measured the mating frequencies. The results she found were when the flies from the starch population were mixed with the flies from the maltose population, the flies tended to mate with like partners. This can concluded that a reproductive barrier had been formed between the two divergent populations of flies, forming new species. So let's talk about sympatric speciation. In sympatric speciation, speciation takes place in geographically overlapping population, which means new species develop in the same area. Now, this can occur in some different. So first we have polyploidy, which is the presence of an extra set of chromosomes in a cell due to accidents during cell division. Now, many think of Down syndrome and chromosome 21 when they think of an extra set of chromosomes, but in some organisms it can cause an entirely new species. Now, an autopolyploidy is an individual who has more than two chromosome sets that came from the same single species. Then we have alloploidy, which is when different species mix together multiple sets of chromosomes. So, for example, we have species A and species B, and over time, we get a new species when the two sets of chromosomes are mixed. Now, many important crops today, such as oats, cotton, potatoes, and wheat, are polyploidies. Now, sympatric speciation can result from the appearance of new ecological niches. For example, the North American maggot fly can live on native hawthorn trees as well as the most recently introduced apple trees. So sexual selection can also drive sympatric speciation. For example, sexual selection for mates of different colors has likely contributed to sympatric speciation in cichlid fish in Lake Victoria. Now, sympatric speciation resulted when the cichlid fish choose their mate during sexual selection. So different species of the cichlid fish do not mate based on the color, so in the same environment under normal light, they will not mate. But if you put them under the same orange light, the two species will appear identical and they will freely mate. So, based on sexual selection, it causes sympatric speciation. So let's review the two types of speciations. The first one, the allopatric speciation, is when some type of geographic isolation restricts gene flow between the population. And then in the isolated populations, reproductive isolations may then arise by natural selection, genetic drift, and sexual selection. So even if the contact is restored between the populations, interbreeding is prevented between the two species. Then there is the sympatric speciation, when there is no geographic separation, but some type of reproductive barrier isolates a subset of the population from the parent species, resulting in a new species in the same location. Now, sympatric speciation can result from polyploidy, from natural selection, and from sexual selection. So let's talk about what's called a hybrid zone. A hybrid zone is a region in which members of different species mate and produce hybrids. Hybrids are the results of mating between species with incomplete reproductive barriers. So a hybrid zone can be a single band of land where adjacent species meet. For example, two species of toad, the fire-bellied toad and the yellow-bellied toad, interbreed in a long, narrow hybrid zone in Europe. So here's the hybrid zone where the two adjacent species interbreed. Now, often hybrids will have reduced fitness when compared to the parent species. The distribution of the hybrid zone can also be more complex if the parent species are found in patches within the same region. So now, when closely related species meet in the hybrid zone, there are three possible outcomes: reinforcement, fission, and stability. Figure 24.14 shows the formation of a hybrid zone and the possible outcomes for the hybrids over time. So when species meet in a hybrid zone and reinforcement is the outcome, the species create hybrids that are less fit than the parent species. So over time, the rate of hybridization decreases. Now, where reinforcement occurs, reproductive barriers should be stronger for sympatric than allopatric species. So barriers are stronger for species that live in the same area than for species that live apart. For example, in populations of flycatchers, males are more similar in allopatric populations, ones that are separated by geographic barriers, than in the ones that are in the same location. Okay? So fusion is another outcome when species meet in a hybrid zone. In this outcome, the hybrids are as fit as the population and there is substantial gene flow between the species. If the gene flow is great enough between the species, the parent species can fuse into a single species. That's why it's called fusion, because they fuse into a single species. For example, researchers think that pollution in Lake Victoria has reduced the ability for female cichlids to distinguish between males of different species, this which has caused the fusion of the species. So stability is the third outcome from the species interbreeding in a hybrid zone. Now, extensive gene flow from an outside the hybrid zone can overwhelm selection and increase the reproductive isolation inside the hybrid zone. So, there are still many questions that remain concerning how long it takes for a new species to form, or how many genes are needed to differ between species. Now, broad patterns of speciation can be studied using fossil records, morphological data, and molecular data. So when studying the fossil record, it is not uncommon for there to be periods where new species appear suddenly in the strata, persist essentially unchanged through several strata, and then apparently disappear. This is what we call punctuated equilibrium. Now, Eldridge and Goldie came up with the term punctuated equilibrium to describe these large periods of no change punctuated by sudden change. The punctuated equilibrium model is caused by sudden changes such as fire or a disease outbreak. Now, the punctuated equilibrium model contrasts the gradualism model in a species' existence. So here are the two models. The first model is the punctuated equilibrium model, which is when speciation occurs rapidly and a new species changes most at the beginning as it buds off from the parent species and then changes little for the rest of its existence. Then you have the gradualism model, which is when a species descends from a common ancestor gradually. The new species slowly diverges more and more in their morphologic and as they acquire new adaptations. So the punctuated pattern in the fossil record and evidence from lab studies suggests that speciation can be rapid. For example, a sunflower species came from a hybridization of two other sunflower species. Here's a picture of the hybrid sunflower species and it on its habitat, a dry sand dune. So the intervals between speciation events can range from 4,000 years in some cichlids to 40 million years in some beetles, with an average about 6.5 million years. So the fundamental question of evolutionary biology that still persists is how genes change when a new species forms. Depending on the species in question, speciation might require the change in only a single allele or the change in many alleles. So, last thing, macroevolution is a cumulative effect of many speciation and extinction events.